Thank you to the organizers for inviting me to speak. So today I'm going to put on my Arabidopsis hat. For those of you that don't know me, I work on a number of different plants. Tomorrow I'll have the opportunity to tell you about the work that we've been doing in rice. But in one of my other roles, I'm the interim director of a consortium that is focused on Arabidopsis informatics and trying to figure out where the future is and how to address future problems with Arabidopsis informatics. And so we call this the International Arabidopsis Informatics Consortium, the IAIC. And I'll tell you about a portal that we've been discussing that will hopefully address some of the upcoming problems in Arabidopsis informatics. So just to give you a little bit of, and I should say that the other reason that I'm discussing this today is I think that there's perhaps an opportunity to learn from what's been going on in Arabidopsis and learn from how we're addressing the problems of dealing with the massive amounts of information, and there may be an opportunity to apply some of those lessons to rice in the near future. So what we face in the Arabidopsis community is an issue of rapidly growing amounts of data, whether it's genomic data, genotyping data from 1,001 genomes that are being sequenced, whether it's transcriptomics data, metabolomics data, proteomics data, phenomics data, just an incredible amount of data that's coming out. How can we consolidate these, integrate these, and make them broadly available? And of course, individual investigators, we all have our areas of interest, and we're often both data generators as well as data consumers. And so it's really important to have these tools that we develop in our individual labs for handling these specialized data widely available. And then the other issue that we face is that the Arabidopsis community is really a global community, and yet a lot of the informatics support have been provided on a national level. The U.S. National Science Foundation was pretty much the sole supporter of TARE, the existing Arabidopsis information resource, and really the use of TARE is only about 25% from the United States. So the question is, since about probably 90 to 95% of the funding was coming from the U.S., but only about 25% of the use, the Science Foundation was interested to know how we could grow the sources of funding, perhaps leverage their funding across borders to make a more international resource. And the Arabidopsis community really wants to enhance its role as a model system in a way that to take advantage of these informatics resources and efforts to develop new tools to keep Arabidopsis in the top tier of model organisms. And so if you're not familiar with TARE, TARE stands for the Arabidopsis Information Resource, and it is really the previous and current informatics resource for Arabidopsis. And it has a relatively complex design, a centralized data source, and it was really designed to be a central data repository and central source for data. But data is less and less centralized, and the resources are less and less centralized in the tools. And so part of the issue was for the National Science Foundation was again trying to internationalize and distribute the effort, and so TARE's funding in the last funding effort, it was decided that this funding would be gradually phased out such that in the summer of 2014 it would no longer be funded. And part of the reason behind this was not that the U.S. National Science Foundation was any less interested in funding Arabidopsis informatics. It was just that they wanted to come up with a new way of approaching the problem and new ways to spend their money so that they could get more bang for the buck. And so we saw this then as really an opportunity to sit down and think about how we handle the data and how we can really address the growing bioinformatics needs in the plant sciences over the coming years and come up with some sort of a system that will be sustainable both scientifically to deal with these massive amounts of data as well as financially over the next 10 to 15 years. And the U.S. National Science Foundation has been quite supportive of this effort, even though they've essentially marked TARE for an end in 2014. At the same time, they've really been supporting the efforts to think about where we're going next. And so what we were thinking is it's really the time to be innovative, far-reaching, inspirational, and to set up a model for handling plant information and plant data that could serve as a resource and an approach that could be used for other organisms, perhaps such as rice. And so the NSF support for this came in the form of a grant. This is about $500,000, what they call a Research Coordination Network grant to set up this consortium. I was a PI on that, and then I have a number of co-PIs, Doreen Ware, Eric Grotwald, Volker Brendel, Dan Stenzioni, Ryan Dooley, Jim Carrington, and then we have a number of international steering committee members as well. And so we really sat down a couple of years ago and started to think about this. That led to the award about 18 months ago that we've been using to fund this effort. So let me give you a bit of history about this. So really these discussions started with a meeting set up by MASC. MASC is the Multinational Arabidopsis Steering Committee. This was in April of 2010, and it was followed up about a month later with a second meeting that was coordinated by NASC, which is the North American Steering Committee. And these groups, there were about 50 participants in each of these groups, so it was really meant to be a community thinking effort to think about where we needed to go with this. This led to the establishment of our consortium to coordinate these efforts, and then we've continued the discussions both at the Arabidopsis meetings and at the plant and animal genome meetings. So we tend to meet about every 6 months in person to discuss this. The initial work led to a publication in the Plant Cell, and just to put in a plug for Plant Cell, they've been very supportive of this effort and have published not just this paper, but I'll show you another one about a year or two later. And so this initial paper really set out the thinking about the consortium. It was a result of those first two workshops that were held in early 2010. And we came up with a structure and that would have at its core this portal, the Arabidopsis Information Portal. That portal would support directly efforts including the stock centers, so keeping records of what resources are available, the gold standard genome annotation, as well as curation of functional data. And so this portal then as a central hub would allow the sort of plug-in of modules that would support examination of the metabolome, the epigenome, ways to predict phenotype, predict function, protein interactions, so on and so forth. But these various modules might be developed in individual labs and simply access data that reside in the portal and contribute that data as well. And then there's the potential to allow easy access for comparative genomics, including to other plants, potentially including rice. And thus this portal, this AIP, represents the primary interface which provides us access to these core resources, indicating these resources up above, as well as non-core resources such as the additional data types, and as well as links out to boutique databases, which could include individual labs, databases, and resources. So it's a central hub for coordination of Arabidopsis informatics. The timeline for this we set up at the beginning, so this was set up at an ICAR meeting in Yokohama in the middle of 2010. We are now at about this point, so TARE funding is rapidly decreasing, and then we came up with a plan to develop these resources over several years with the goal of having core funding by 2012. We're a little bit behind this, but I would say that many of these efforts are well underway, and so we've coordinated the development of the portal and the consortium. We're working with investigators in different home countries that are starting to write proposals for funding in their own countries, which can plug into this central portal resource. And part of our effort in thinking about this was really a kind of a group thinking effort that we had about a year ago now in Atlanta. So we got about 50 people together. This included plant biologists, cyber infrastructure experts, bioinformaticians, members of funding agencies, and we had a professional coordinator who works on these types of thinking exercises. In the next few slides, I'll tell you about what the outcome of that design workshop was. But this was an incredibly important exercise because it was a community effort to think about where we'd like to be in 5 years and in 10 years, which is relatively difficult not knowing what data sets will be available. And so the things that we are thinking about are, what is it that we need to build? How are people going to use this? So what are these use cases? How do we make this broadly accessible to biologists, to bioinformaticians? How do we integrate extremely diverse data types? And how do we make this robust to the rapid changes in data that are taking place? And then, of course, how would we actually build it? How would we identify computer scientists who would be able to design this and carry that out? How would we fund this? And what is our action plan in order to implement these ideas? Again, with the aim of coming up with a plan that will keep Arabidopsis at the forefront of model systems over the next 5 to 10 year period. Oops. We set up, ahead of this meeting in Atlanta, we set up a series of working groups that focused on the 3 major areas that we believe needed to be addressed in the construction of a portal: an engineering, architecture, and infrastructure working group, a modules and use cases working group, and a standards and ontologies working group. And so we had experts. We had experts both leading these groups, and we had participants from many different countries that were involved in the process, and we tried to break the group up, the larger group of 50 or so people, into one of these 3 categories, and they met ahead of time with virtual meetings using Skype and other tools to come up with ideas ahead of the time. We also sent out surveys to various existing model system portals and databases to discuss the different types of community, social challenges and approaches, and technical challenges that they've faced in their experience in building these portals. So what did we do? This is a 2-day meeting. The first day meeting was really spent sort of getting everyone up to speed, and then the second day was really this thinking exercise and trying to imagine 10 years from now, where do we need to be? What types of data do we need to deal with? What's the scale of that data? And how are we going to interact with those data? If you think about the changes in technologies, think about your iPhone or your mobile phone and what that technology was 10 years ago and what you would have thought the technology would be today. It's really dramatic, the amount of change that can occur over 10 years. So we were thinking, realistically, how are we going to get there? And then we had a number of group exercises to think about how we might design this system and what the vision might look like. We had a lot of sketching and interactive groups and came up with some really interesting ideas about how this portal would be structured. In other words, it needs to be interactive and social. We need novel visualization tools. It needs to be highly customizable, highly accessible data so that anyone can grab those data and work with them. It needs to be elegant and professional, and it needs to enable in silico experiments so that you can do predictive work, predictive biology, trying to understand what sort of phenotypes you'll have if you modify one gene, several genes, entire pathways. It also, on the back end of it, it needs to be open, powerful, professional, robust, reliable, and very well curated. And so we came up with various what we thought were the most transformative aspects of this portal. It needs to have this modular design to integrate global resources. In other words, we need to have some common core which may be centrally funded and centrally built, but we really need to make it accessible to individual labs to contribute tools, to contribute data, and to get tools and data back out of it. It needs to have a sort of dashboard where you can easily access all of these tools. You need to allow extreme curation by the members of the community. We need to reach out, in other words, for the community to get all of their expertise and pull that in as easily as possible. We need to allow full integration of social media tools, and you're probably familiar with apps from your phones, for example, and a lot of young scientists are really quite comfortable with the social media types of tools, Facebook, so on and so forth, and so we need to encourage that, to encourage young scientists to collaborate across borders. And we need to allow direct access to the database so that power users can really get in, get the data that they need, get all of that data, get all of the metadata about it, and work with those data at a very fundamental level. We identified a number of challenges. How do we get the community to buy into this, to support it? How do we get the community to agree on the standards for data and to adopt those standards in the generation of their data? We recognize that software development is expensive. How do we get around that, or how do we work with that as a constraint? We need to direct community activities toward the development of these applications to handle the data. In other words, right now each lab may have their own systems for dealing with the data, but how can we convince the community to work with those algorithms to put them in a context that can plug into this centralized resource? We also need to figure out ways to deal with the massive amounts of data that are coming out, and we need to leverage existing tools for cyber infrastructure. All of these thoughts were put together and put into a paper, again, that was published in Plant Cell because of their willingness to work with us on this. So we have a commentary, taking the next step, building an Arabidopsis information portal. It came out in June, and I encourage you to read over that to get an idea of what our thinking was. And a lot of the ideas that I'm presenting today will be found in that manuscript. And so in order to direct the development of this portal, the steering committee got things going. We now have a scientific advisory board, an international board with elected members that's facilitating the work, international work, to develop these teams who are of people who are interested and willing to develop parts of the portal. They've identified a number of possible funding mechanisms and really are trying to encourage participation by members of the Arabidopsis and other communities to develop these resources. And then really the next challenge will be to develop modules that plug into this. And I'd like to just say that Chris Town stepped forward. He's at the J. Craig Venter Institute in Maryland, submitted a proposal to the U.S. National Science Foundation to take the first step in developing this portal. And so how are we going to get there? How are we going to get from where we are today into the future? And so really the first thing that we need to do is deal with the loss of TARE, which is a big blow for the Arabidopsis community. A lot of people are very upset about it, but we do have a plan in place. Rod had mentioned a few moments ago the iPlant Collaborative, which is a NSF-funded cyber infrastructure project, which includes both hardware and software to support plant biology. And so the current plan is that the resources that exist today in TARE are being transitioned. So TARE will not go away. It will just go into a static form in which it's not updated. And so those will get migrated from their current state at the Stanford University, at the Carnegie Institute, and migrated to iPlant. And then from iPlant, as the portal is developed, the existing data that are in TARE will get migrated into the new portal. And then as individual investigators, individual labs start to develop these modules, they may require additional data that will come from TARE, and hopefully they'll be developing new data sets that may not have existed in TARE, but will find a home within the portal. And so what then is the structure of this portal? If you're a computer scientist, how will it be developed? Well, Chris Town had identified Intermine, which is a database system which is developed to integrate data for Drosophila, has about 100 person-years of development behind it. It enables the collection of large data sets. These can be loaded into the database using a variety of different parsers. It has an object relational model that allows the connections between very different data types based on common identifiers, and it gets rebuilt and re-indexed on a periodic basis. And so Chris has come up then with an architecture for this, which takes the legacy data, which is coming from TARE and other sources, we'll migrate this into a new database. We'll have a large amount of bulk data storage, which is provided by iPlant, as well as processing backends. And then we'll have web services, which will grab the data from the existing data, as well as new data that might come in from various sources into the bulk data storage. And there'll be a variety of different graphical user interfaces for biologists to view those data. And so we'll also, this resource will have ways to parse external data sets, and ultimately we really want these modules, the user-developed, community-developed resources to come up with different graphical user interfaces and different ways to connect to those data and relate to the data which will exist in this Intermine instance. And so Nick Provart and colleagues have done a great job of thinking about different use cases. In other words, different ways that biologists like you and me might interact with these data. And so this is where the idea that different user groups, that people that have interests in alternative splicing, epigenetics, metabolomics, protein interactions, genome diversity, will come up with different algorithms that can be applied to the data, but we really need to be able to reach reach into the public data set, grab those data easily, work with them, and then we also want to make our tools available to the public so that we can all leverage the best resources that are out there. And so we came up with a list of action items that are actually we're underway with many of these. Coordinated development of the portal, community participation in the oversight of this consortium via a scientific advisory board and a scientific advisory panel that will provide an independent assessment of the progress that's being made. We need to continue to develop the standards for data, particularly as data continue to evolve. Push for open data, really excellent metadata to understand where those data came from, how the data were generated, open source requirements that need to be enforced. And we also need to encourage proposals for different modules, different tools to be developed internationally so that they could all work together in this federation of closely knit components that will comprise the larger international portal. And then we also need to push our own national governments to support through core funding potentially, so this is more like long-term funding, the core resource of the portal. Okay, so how does all of this relate to rice informatics? I've told you what's going on in the Arabidopsis community. I do have a strong interest and worked for many years with rice as well. And so these are just a few of my own ideas about how it pertains, but I'd be really interested to hear yours. But I think there's maybe a parallel need then to come up with a portal for rice that could develop based on many of the same ideas that are going into the Arabidopsis portal. So perhaps we need an information portal for rice. So just throwing out a few possible acronyms, the IPR, and this could be shaped by a consortium made up of many of you in the audience who might have an interest in this, an International Rice Informatics Consortium. However, Arabidopsis may be somewhat easy in some aspects, which is that there are really no economic interests at play in Arabidopsis. It has a long history of international cooperation. Funding has come from, a lot of funding for Arabidopsis work has come from the BBSRC, the European Union, the U.S. National Science Foundation. All of those agencies mandate open access to information and resources. So from the get-go, all of us that are funded by these various agencies have to ensure that our data and tools are open and freely accessible. And there exist Arabidopsis stock centers which distribute these materials worldwide as freely as possible and try to help researchers work with the MTAs and know what all of those terms and conditions are. Major areas in which rice may differ from Arabidopsis is that many of the resources are a bit more fractured in that it's not always clear what resources are freely available or not freely available, or how we can access them across borders. And so there can be some problems sharing resources internationally, whether it's germplasm or even data. Some funding agencies don't value the free sharing of data and resources, so it may not be mandated that researchers have to make this available, and so that can slow down the process of sharing that, and that in turn is unattractive to people who are perhaps not working directly in the rice community but are right on the border and may be interested to do some work in rice but can't get those resources quite as easily. Maybe they're doing comparative genomics, maybe they work on maize and they want certain mutants that have orthologous mutants in rice, and it may not be so easy to get them. And then intellectual property and barriers to access may be increasing, and this also causes problems as well. And then we need to think about what we as a community, putting on my rice hat, what we in the rice community might want. In other words, what type of data sharing agreements would we like to have? How can we use these to access the resources more freely? And so far we've had many really well-funded public projects that have been contributed to this greater good. If we have a closing of resources, the contribution of those resources to the greater good of improving rice may diminish over time. How also can we organize this rice portal to provide access to the data? That data may include diversity, germplasm and genotyping data, phenotyping tools and data, and resources and information that are coming from all of the major rice projects that are represented here or not represented here. And then this would allow us to really facilitate and streamline access to the data if we had all of this, access to the tools, access to the resources, including those genetic resources. And it would also be helpful if the terms or conditions of the MTAs or specific agreements could be much more transparent, explicitly outlined in some sort of a central resources, so that if there are barriers to getting those resources, we know exactly what those barriers are and how to work with those. So then, if you're interested in developing this portal for rice, we need to think about what it requires. And really, I think international collaboration and cooperation. In the case of Arabidopsis, we've spent 2 years so far developing this collaboration and cooperation to come up with a plan for how to do this. And potentially in rice, perhaps this is something that could be coordinated by IRI, perhaps under this the Global Rice Science Partnership that was already mentioned once today, GRIS. So then just additional questions that we could discuss. Do you think as a community that there is a need for a public portal, publicly available portal such as this? What type of information should be included? What are the priorities that we have for this? What components already exist and could be leveraged? Who is the primary audience for this? And really, most often as a biologist, I think it's the biologists who need this. But are there prototypes that exist that could allow us to move more quickly? And I think what's happening in Arabidopsis may exactly represent such a prototype. So finally, I just want to mention that we have a web page, arabidopsisinformatics.org, and we have various updates that come out usually about every 6 months as we have these meetings, either the plant genome meeting or the Arabidopsis meeting. We will have another workshop on this at the plant genome meeting in San Diego in January, so I encourage you to join us then. So thank you very much.
Did you, I might have missed it, did you give us any idea of what the budget is for to set up such a system? I mean, I saw you put the grant in.
Right. I don't actually know what the budget was on Chris's proposal, but in talking with the funding agencies and some of the earlier discussions, we had come up with a price tag of around $10 to $15 million over a 5-year period, so about $2 to $3 million a year, and that I think would allow us to develop this central resource, which would provide a home for the data where everybody could deposit their data. So sort of data warehouse, as well as the centralized components, the architecture that would allow people to plug their resources into it, a staff, the development staff, user interface, which would provide some of the more important data, access to some of the more important data. I think the genome annotation, updates of that genome annotation, access to the stock centers, curation of functional information. But then we also envisage that there will be additional funds which will be contributed sort of in kind by individual researchers, bioinformaticians, biologists who are developing the tools in their own labs, which would be, they would benefit because by plugging their modules into this centralized portal, people would become familiar with their tools, they would get recognition for their tools, and they would get free access then to the data which is in this centralized resource. So perhaps there would be an additional $10 to $15 million in sort of in-kind contributions through the development of these modules that would be funded by other funding agencies internationally, as well as the funding agencies which are providing this core resource.
Okay.
Blake, thank you for a very interesting and very important presentation. I know that you sort of drifted to this at the end, but is there a similar discussion going on in the Arabidopsis community about the genetic resources, the seeds and the mutants? You've talked mainly about the databases, and all of us were shocked when we heard that the funding is also being lost for the distribution of seeds and things. So are there simultaneous discussions? Because, that's all part of the picture.
Right. Well, I'm not quite sure that I know what you mean by the loss of funding for the distribution of resources. The ABRC, which is the primary distributor of seeds in the U.S. and does a lot of international distribution, is quite well funded and has been renewed recently. So there's no concern on the distribution side for Arabidopsis about the continued investment of funds. So that should continue. And the ABRC spends a lot of time making sure that that the resources that they acquire for distribution are as free of MTAs as possible, and working with the people that are contributing those resources to mitigate any distribution issues, because for them as a stock center, it's a real hassle to have to deal with different agreements for different materials. And so in their ideal world, none of the resources that they want to have or want to distribute would have any MTAs associated with it. You could simply go on the internet, click on the seed packet that you want, and they could send that out. They also spend a lot of time, of course, dealing with international agencies, USDA, for example, or APHIS, to make sure that they have all of the licenses and permits to do that. And so I think having those types of efforts centralized, so that as scientists we don't have to worry so much about dealing with the MTAs, dealing with our own government regulatory agencies, and we can simply work with one set of papers in order to make sure our lab has the permits that we need, and then everything else flows. Build out this Arabidopsis information portal. That should really sort of pick up where TARE left off. There will be a gap which I plan to stepping in to help fulfill, but NSF has in no way stepped back from funding Arabidopsis informatics or resources. And so, in fact, they were the ones which encouraged this discussion. They funded the $500,000 coordination network grant in order to think about where we're going next. So
I think it's a risk to count on a funding agency like that for long-term funding of any biological resource, because they are not supporting long-term anything other than Arabidopsis, except for maybe Drosophila and other things. So I think we have to think beyond just scientific funding agencies that work on a 5 to 10 year grant. I think it has to be a much more solid funding base.
Right. I mean, that would be nice. In the U.S., there are perhaps few options. But I think as long as we really spell out the importance of the work that we're doing, and hearing Rod's talk about the 9 billion person question, I mean, questions and issues like that are of really fundamental importance, and so I don't see how they can't fund us.
All right.
I hope you're right.
All right. Thanks, Craig.